we have a tradition at this meeting of having 2 keynote speakers, and this year we're continuing that. I'm particularly excited this year with the 2 speakers that we were able to secure for this event, and that they were so kind as to donate their time for this. Tony and I both thought that it was really apropos, and then Karen came along and joined in as well, since the recent licensing of venetoclax, and it's now being used in patients on a regular basis. I know the clinicians that I work with, it's now part of the armamentarium for treating CLL patients, that it was an opportune time to look back over the BCL2 family and the drive towards the clinic and the translation of the work that we've been all dedicated to for quite a long time. So tonight we have as our first keynote speaker Craig Thompson, and I have Tony on my no senders list for my email, so I didn't find out until dinner tonight that I'm introducing Craig. So I went back to my room to try and look up what he's done and realized that at 10 o'clock tonight I might have gotten through the list. So instead, I'm going to just tell you a couple of things I remember. He trained as an MD, and many of you may know he was in Chicago and then Pennsylvania, and now at Memorial Sloan Kettering, where he's CEO and president. So it's really great that he offered the time to come today. What I remember most is that in the early days, he was one of the first people really tearing into the BCL2 family proteins, cloning BCL-XL, seminal work on BAX and BAC and the BH3 proteins, but at the same time, always thinking about the potential for clinical application. And I remember one particular presentation where he was talking about IL-3 dependent cells, and that when they were expressing anti-apoptotic proteins, they couldn't die, but they shrank. And that meant metabolism. And really, Craig pushed this whole field into a new appreciation for metabolism, and many other cancer researchers in that direction as well. And it's been really an amazing explosion, as all of you know now, in more recent times. So I'm not going to keep going on. I'm going to let you have the floor. That's right. Thank you a lot.
Thank you, David. I think this is on now. So it's, I really want to thank David and Tony and Karen for inviting me. Tony gave me instructions, and I'm supposed to be the old guy that gives the history of the field, so I'll try and do that. And then at the same time, I'm the setup man from Andrew, who will give us the amazing story of how a drug actually came into the clinic to benefit patients and be approved by the FDA and soon worldwide in many venues, which is an exciting landmark for this field. So I was going to take you back through how this meeting started and how this field started, take you how we are going on to the exciting questions that are going to be addressed here over the next 3 or 4 days, and why the meeting is appropriately called cell death rather than apoptosis, but we're going to talk in this first session about really the role of apoptosis as a founding part of our broader understanding of cell death. So I guess I was telling guys at dinner tonight, this was a pivotal meeting. The 4th of the meetings that you're attending right now took place in 2001. Andreas probably remembers it. It was right after 9/11. We were actually supposed to meet the week after 9/11. We got canceled. We came back together as a community because people believed that cell death was still something we needed to do, and we came here into this meeting in that November. 2 months later, everybody had to rebook, but the place was packed. And we were isolated here on the island for 4 days because a plane blew up on the second day of the meeting that was flying out of JFK. In that case, it was one of the early new Airbus liners that had a problem with its fuel tanks and blew up, but everyone thought it was a terrorist attack. So it was one of the really dynamic times in the field. So I showed this slide from the cover of Cell at that time because it was the change in the paradigm for who were the death effectors, who were the death survivors. And this was the order of the BCL2 family at that time. So I'm going to tell you how we got there and how we got from this cartoon to at least thinking there was something that could be done in the clinic in the next 45 minutes. So that's the introduction. Cell death came from three fields, really. The first field is not one that's broadly appreciated in this audience. The first time we came together as a community to be the history of this was actually a Banbury conference that was organized across the bay here at the Banbury House, because there were so few people that were interested in working in the field. The biggest argument at the meeting was how to pronounce apoptosis. And three communities came together. This is actually, in my belief, although I should say, I took every name out of the slides so that no one can be blamed for this history other than me, okay? I'll try and mention people when they've done things, but I will undoubtedly inflame some of those that have been part of this community now for the last 25 plus years. And so the three communities that came together in that very first cell death meeting that took place here at Cold Spring Harbor was a community that was the cell biology community. The cell biology community had been worrying about programmed cell death, they didn't quite call it that, since basically the 1930s. It really became galvanized in the 1940s when people tried to culture your favorite cell type and weren't able to do it. So that's when HeLa cells were born, the only cell line everybody can grow, because it grows in water as best we can tell. And a variety of ascites cell lines that had to be passaged in mice. And every time people took them out and tried to grow them in culture, they went through this morphologic change. They went from being happy cells, growing happily and continuous in a serum-derived media or in ascites fluid, to undergoing this morphologic change in about 48 hours, which is what we know about apoptosis. And when they were characterized, all the DNA was degraded to oligonucleosomal ladders, and the cell had just simply auto-digested itself. But the cell biological community basically missed the boat. They knew this happened, and so starting in 1948, Victor Hamburger and his colleagues in WashU started looking for the factors in media and serum and ascites that kept the cell alive and avoided this. And so their whole field launched growth factors. That's how NGF was discovered by one of the postdocs of Victor, and everything went on to discover growth factor signal transduction. And we would have been stuck ignoring the death problem, the death by default that cells undergo when they're taken out of their norm physiologic environment and not signaled by growth factors, except that there was another, probably more public, that came from anatomy. And this is the example I like, because at the time my lab was working actually on chickens. And so there was a great deal of evolutionary biology or developmental biology that was done in the field of avian work, because chickens were oviparous animals, and you could look at and watch their development through a hole in the egg and see every stage of development. And one thing that was striking as an example that programmed cell death was important was that chickens and their closely related species, duck, formed a whole foot pad. So it was one syncytial tissue. And then, once fully formed, two entirely independent events occurred, which is that the chicken involuted all of the tissue in between the digits so they could be completely separated, because a chicken hunts for its food by pecking in the ground and by clawing, defends itself with its claws and its talons. And at the opposite time, the same exact primordial structure in the duck becomes highly organized to be webs that allow it to be effective as an aquatic animal. And the difference was death, the developmental death that occurs to get rid of this tissue, which is prevented in the duck. So some people still thought that that was a growth factor dependent phenomenon. Other people thought death might be interesting. And that led to one understanding as people got into developmental models and could use genetics to study these problems. That led Bob Horowitz and his colleagues, as they characterized the lineage of C. elegans, to understand we could address that question in C. elegans by doing a genetic screen. And they were able to show, beyond a shadow of a doubt, that in fact this was cell-autonomous death. It was not the prevention of death by other things, but cells that were actually dying with a self-instruction program. And they had characterized, at the time the meeting had come together, a series of genes, the SED genes, cell death effector genes, that were required for this. 2 that were required for affecting what we came to know as apoptosis. Apoptosis came from the pathologist community, was recognizing the cell death that occurred in tissue culture, also occurred under the normal turnover of cells in the liver, and so that people thought of this as a normal physiologic turnover and the cell distributing itself, but Bob showed that there was genetics to this. There were 3 genes, 2 genes that were required to affect it and 1 gene to oppose it, and present in all cells. And I'm not going to go through any of the beautiful genetics they did there. We all came together as a community. The other way that sometimes genetics more and more informs biology, though, is that human beings are collecting disease syndromes. And we now have a human genome, but at the time we had various ways to analyze whether there are genetic abnormalities in humans. And in cancer studies, it had been discovered by Carl Croce's group that there was a reciprocal translocation in a certain type of lymphoma, B-cell follicular lymphoma, in which 90+% of the patients had this reciprocal chromosomal translocation. And so an arm of chromosome 18 switched places with an arm of chromosome 14 and created this breakpoint neogene, which was a fusion of the immunoglobulin heavy chain with a gene that became known as B-cell lymphoma-associated gene 2 in Carl's nomenclature, or BCL2. And so that launched an understanding that cancer also could discover genes in essentially a genetic way and inform another pathway. And at the time that we all came together in that Banbury conference, it had just been described, first by David Vo and then by Stan Korsmeyer's laboratories, that this abnormal gene created by fusing the regulatory parts of the immunoglobulin enhancer to the coding parts of BCL2 led to the production of a gene that played no role in anything that was known about oncogenesis at the time, because it didn't cause cells to proliferate. It didn't act like a tumor suppressor in that it was mutant acted as a dominant negative. It simply kept cells alive and reversed that cell biological problem that the cell biologists had known for years. When you took cells out of serum growth factors, they no longer knew how to die. And so that was a really important shift in cancer, and it's why this story, from my talk to Andrew's talk, will keep coming back to cancer. For the first time, the community had to face that for cells to accumulate as a mass didn't just involve the activation of oncogenes discovered by avian retroviruses, and by and large had the collective properties of capturing signal transduction to initiate the transcription of genes necessary for the cell to enter the cell cycle and divide, or remove tumor suppressors to prevent at checkpoints of the cell cycle the effectiveness of that drive to proliferation. That would get you more cells, but ultimately, if there was a balanced form of cell death, you'd get back to homeostasis. And it led to the idea that you could dysregulate cell survival signaling or genes involved in the genetic execution of cell death, and that could lead to a carcinogenic process. And so we came in, as David just said, when we, in studying chickens and bothered by the fact that it was really hard to culture the cells we were interested in, decided just to borrow from Stan Korsmeyer the idea that we could introduce a gene like BCL2 and keep the cells alive long enough to study their physiology. So all our job is to clone the chicken homolog of BCL2. And I will admit, it started on an August morning when I went on vacation and gave my technician the job. We just laid out a library. I said, here's your probe for BCL2, go clone the chicken homolog. And came back from vacation, as all good experiments are done, and she said, I cloned BCL2. And I said, that's great. And it had lots of homology, and we could show it at all the things. But as I was saying, let's just get concentrated. We'll put this into this vector we're interested in. She said, but you know, I cloned something else. And I said, what do you mean? She said, well, there's this other gene, and it looks a lot like BCL2, but it's clearly not BCL2. And so that became our interest. We had another gene. This was now maybe a family. We called that gene BCLX because we didn't know what it did. We actually knew at the time that Stan Korsmeyer had cloned another gene, homologous, and I sort of said, well, if they have the same gene, then we're not going to work on this. We'll just do our avian experiments. But we exchanged sequences. They had BAX, and we had BCLX, And that allowed us to start this wonderful exchange of reagents between our laboratory that allowed us to work on the various forms of BH2 and BCL and various different forms of BCL2 over the next 15 to 20 years. So I'll talk about that. BCLX, once we had it in hand, Larry Boyes is here. He was the one who was forced to do the full-length clone because he was a postdoc. I still wasn't going to do all this work. And this is the experiment that also David mentioned. We just put it into a cell line that we loved. We worked on hematopoietic cell lines. These are all IL-3 dependent cell lines, immortalized but not transformed. And if you put BCLXL in or BCL2, I just didn't put that slide in, and now take away IL-3 from the growth media, every cell dies in 48 hours. In fact, that first slide is exactly this experiment. Those are the control cells. They all die by classical apoptosis, all the biochemistry you know about. But if you had BCLXL in, 9 days later you could come back and more than half the cells were alive. But more importantly, what Larry showed was you add back IL-3 and every one of the cells that's alive recovers to proliferate. So this is meaningful cell survival. That's what these genes do collectively, not just BCL2, but BCLXL did. Now, the odd thing is that Bax did the opposite in an abnormally spliced form that some people may know. BCLXS also reversed this property, and we were off to thinking this might be a family that regulated the cell death genetic decision. But the critical point came when we convinced Cold Spring Harbor to go from a Banbury conference to being in this room. And so this picture was actually taken at this room. And this was the transformative event of the mid-90s because, in fact, this guy right here, Michael Hengardner, I don't think has come today, was the postdoc in Bob Horowitz's laboratory who was assigned to identify what SED9 was, the inhibitor of cell death genetically in the worm. And he and Stan are talking. You can see how old this is because he's got slides. Stan doesn't have a PowerPoint. He has slides right here, okay? They were just exchanging the first information about the fact that BCL2 and SED9 are the homologs of those two species. And so suddenly we knew that this was a genetic paradigm that carried out throughout the metazoan lineage. And so there was the ability to understand with the power of genetics in the worm the different regulators and to compare them to the elegant regulation that you all know about in mammalian species, which took place over the next 10 years. And Stan and Bob, Michael's mentor, were the actual organizers of that meeting. So what came out of that paradigm in the worm? SED9's the BCL2 homolog. Mammalian cells, we found the same gene by chromosomal translocation. SED3 ended up being the caspase effector of cell death. And caspases were subsequently, I think Don Neumeyer's here, he and his colleagues found caspases that allowed us to identify homologs. It's an expanded family, as we now know, of genes that affected apoptosis. And so the genetic signatures of these molecules were all the same. And then the field went to serious work, as David said, which is to try and understand not just the genetics, but the biochemistry of how this effector function works. So to skip ahead, we in our laboratory have always used the paradigm that is central to the BCL2 family, which is growth factor withdrawal. So I'm just going to keep going back to that IL-3, but it's really a common pathway in many forms of what are called mitochondrial or cell-intrinsic cell death. When growth factor signaling is lost, the biochemistry that was elegantly done by Xiaodong Wang and his colleagues led to the discovery that the molecule that activated caspases, intracellular mammalian species, the SED3 homologs, when he purified biochemically to homogeneity, was cytochrome C. He then put the cofactor that was required to activate the caspases and purified that, and that was a cytosolic heptamer known as APAF1, but APAF1 only became capable of activating the caspases in the presence of ATP, so a cell that still has bioenergetics, so this is the opposite of necrotic cell death. The cell actually has to have energy to affect organized cell death. And at the same time has lost cytochrome C from its intermembrane environment where it carries electron in the electron transport chain out into the cytosol, which should be an irrevocable event for the cell to commit suicide. And so that became the pathway and the caspase activation cascade and the various components downstream that led to cellular autodigestion were characterized. So what was left in this was for the BCL2 family field to figure out where the BCL2 proteins worked in this paradigm. So the clear end effectors were the caspases. The clear amplifiers were the things like SED4 or APAF1 and this biochemical intermediate that did not come out through genetics, cytochrome C. But what did the BCL2 proteins, how did SED9 inhibit this in the worm? How does BCL2 inhibit this in mammals? Well, the first paradigm, I think, really came, at least in my way of thinking, from Stan Korsmeyer's laboratory, where they identified this really peculiar property. I just talked about signal transduction, right? That goes from the surface to really elegant places like the nucleus. But ultimately, this, because of Xiaodong Wang's work, had to end in the mitochondria. And when Stan and his lab characterized where BCL2 proteins were, they were all stuck on the surface of internal membranes, the outer mitochondrial membrane, the outer nuclear membrane, and the membrane of the endoplasmic reticulum. Not the normal place you think of as a component of signal transduction. So again, the cancer community, the cell growth factor community started to wake up and said there's something different about the biochemistry of what this is going on. Furthermore, when you interrogated it a little further, what you discovered in a growth factor withdrawal paradigm, this is again IL-3 dependence. If you deprive cells of IL-3 before they become irreversibly apoptotic, you can recognize that the mitochondria are becoming distressed and swollen in those cells. This is just imaging on a fax. It didn't matter. We had a potent inhibitor of the caspases that came out of the work that was being done at Merck Frost in Canada, and so we had ZVAT as a potent inhibitor, and inhibiting the caspases didn't affect the swelling. But the one thing that profoundly affected the alterations in the mitochondrial function and morphology was the expression of BCL2 or BCLXL at the time. And so that said, they were doing something at the mitochondria to maintain the integrity of the mitochondria. And at the same time, you could take the Xiaodong Wang experiment and introduce cytochrome C into happy living cells and see them execute themselves in 15 minutes. And natural caspase inhibitors like the gene ILP, which I think is now XIAP for those who want to know, could potently inhibit the ability of cytochrome C injected into the cytosol from killing, but BCL2 proteins had absolutely no role. So that isolates BCL2 proteins at the mitochondrial membrane and regulating the integrity of mitochondria at the outer mitochondrial membrane. And that's how the field got focused on this, the discovery of cytochrome C, which should be normally contained by the outer mitochondrial membrane, and BCL2 proteins as regulating the integrity of that membrane. So to try and understand biochemically how that worked, my laboratory had a great privilege of working with Steve Fessick, who was then at Abbott Laboratory. And many groups took shots at trying to get the X-ray crystallographic structure of any of the BCL2 family members to begin to understand it. We now know they virtually all have identical structures, whether they're pro-apoptotic or anti-apoptotic, something that is still not fully explained by the field. But the first structure was interesting. The way it came about was by eliminating 1/3 of the protein. And that was the real surprise to us. We couldn't get, like everybody else, any of the proteins to crystallize or to be observable by NMR structure until we took out 1/3 of the protein, which is what's called the regulatory loop now for people that are aficionados of the BCL2 proteins, replaced it with 9 alanines. And then we got a beautiful crystal structure, which is the core domain of all BCL2 family members, which are 2 central helices that are basically 30 angstroms long, the exact length to go through a lipid bilayer, surrounded by a series of alpha helices. And its closest homologue already existed in the literature. It was diphtheria toxin, which has exactly that. It's a pore-forming domain that regulates the opening of channels and the integrity of the membranes to which it attacks. That's how it actually disrupts your gut. And it made perfect sense because the highly conserved domains that have been identified in all the BCL2 family members that have been named BH1, BH2, BH3, and BH4 by their relative conservation fit exactly that. So these 2 highly conserved BH1 and BH2 are the 2 red helices that make the central pore-forming domain. And the others are the helices that surround it and provided a hydrophobic center so that its hydrophobicity is neutralized when it's in the closed configuration, but can be released into a membrane, and it can be membrane-seeking in its open configuration. I'll say one other thing about the structures of BCL2 because nobody comments on them anymore, and we still don't understand what it means. The most highly conserved evolutionary component of BCL2 proteins is the transmembrane domain. You can tell every BCL2 in the metazoan kingdom by its transmembrane domain. No other domain is highly conserved. Same for BCLXL, same for all MCL1, same for all the family members. The most conserved invariant part is the transmembrane domain. Yet none of us really think about what that does for specifying which membranes the BCL2 proteins go to. We tend to think about the mitochondria, but there's beautiful work that says this inserts in other membranes, including the endoplasmic reticulum, regulates calcium homeostasis, which will be talked about later in this meeting. But this pore-forming domain was well known. So it has a transmembrane chain, the BCL proteins, to localize them to a membrane of interest. And then there were two configurations of pore-forming proteins. When the self-real helixes are protected by the surrounding helixes, like a closed umbrella, and then an open configuration that's driven on all examples that have been tested so far, either by changes in pH or changes in transmembrane potential across the membrane, and that drives the insertion of this domain and forms pore formation to allow usually small anions and cations to exchange. BCL2 proteins are cation selective. So we went out with that insight to collaborate with some colleagues who worked on planar black layers. We had cloned BCLXL, so we decided to study that one. And after a few months of work, we got very beautiful chenilliform activity from preparations of BCLXL. And they would form various opening states based on multimerization and various forms of cooperativity, and they were very highly ordered and regulated channels. To this day, no one has figured out whether that's a useful property or not. Now, I should say, despite everything else I'm about to tell you about the importance of backs and back, you put BCL2 into yeast, which have no backs or BCL2 homolog that anyone can find, and BCL2 functions to keep cells alive from various toxins, particularly ones that are cholesterol solubilizing. Multiple labs have done that. It's a very positive effect. In every cell type you introduced it, from yeast to man, if you put in BCL2 or BCLXL alone, it'll regulate and alter calcium dynamics. So there is some positive function to this, but it isn't important to our story. As I told you, we also were interested in the proapoptotic family members, because by this time, there were a large number of BCL2 proteins. They all had these highly homologous, that all had their membrane insertion domain, and then the BH1 through BH4 domains. Yet, some of the proteins, when you put them into that growth factor withdrawal assays, such as BCL2 or MCL1 or BCLXL that you all know, or SED9, which scores out in exactly the same things, kept cells alive when there weren't growth factors. But others, when you put them in, like BAC and BAX, led to more rapid cell death. And so, even though they had all the same predicted structure, all the same domains, and various domain swaps were done, it wasn't easy to figure out why some were proapoptotic and weren't anti-apoptotic. And so again, the proteins were classified as anti-apoptotic or as proapoptotic, and the two recurrent ones were BAX and BAC. There'll be some talks here about BAC. At the time, BAC was highly lineage restricted, whereas BAX and BAC was expressed in any cell type. So they become the ones that people were most interested. They were present in essentially every cancer cell line we've ever looked at in most normal tissues, as opposed to BAC, which is much more restricted. And so we had made a bunch of preparations of BACs to try and figure out if it formed a channel. And I'm only going to show you one slide, because every time we'd add the preparation, the membrane would disappear. So we'd have this planar lipid bilayer, doing a patch clamp analysis around it. You add in BAX protein, and it didn't work. And we spent a lot of time refining that, figuring out what the problem was. We ultimately got some stable preparations that didn't do much. So we went from couldn't make a membrane that was stable with BAX on it to one that wasn't. And this is really one of the demonstrative experiments I pulled out of the old days. This is a BAX preparation that doesn't porate anything, but if you add what's now known as a BH3 domain from one of the BH3 only activating proteins, it instantly induced chenilliform activity that led over a period of seconds to minutes to dissolution of the membrane. So that led to the model that BAX and BAC could aggregate when tripped by an activator into pore formation. And that's where the field has been, because it's an elegant assay. Once you disrupt the membrane to which Bax and Back are attended, cytochrome C is going to get out, the other apoptotic regulatory factors that have been identified in the intermembrane space. But Bax even itself has nice chenilliform activity until such time as the membrane ruptured, which happened here. So again, that's never really been explained. How does it go from being a channel to something that just porates a whole membrane? That being said, there is no question that Bax and Back activated with BH3s, a number of people from Emily and others that are here have made use of that assay to teach us a great deal of the biology around how it is that Bax and Back lead to the dissolution of the membranes to which they insert. And so the model we have right now is that BCL2 proteins are by themselves relatively inert proteins. They don't have much of a phenotype until they're activated from various signaling pathways that have to do with self-stress by proteins that are known to have a single domain, a single helical domain called a BH3 domain. And when they're either released, as the case that Andreas showed us in BIM from the cytoskeleton, or they get induced transcriptionally by certain stress-induced transcription factors, or they're altered by phosphorylation as a result of changes in signal transduction, they descend on the distribution of where Bax and BCL2 are, the outer mitochondrial membrane, and they have a common property. When they bind to BCL2, they inhibit its function. Another way to think about that same biochemistry is BCL2 sequesters the BH3 domain, so it can't bind to Back. And I would say as a field, we still don't know which is more important, but it's still an important paradigm either way. And at the same time, when it interacts with Bax and Back, it increases their association with membrane, and it converts them to a ability to form formation and aggregate, leading ultimately, as I showed you in that diagram, to the dissolution of the membrane to which they're attached and the release of the contents, the calcium in the ER or cytochrome C in the intermembrane space. And either are sufficiently lethal to initiate apoptosis. The difference is if they do it in the ER, caspase 12 is the initiator caspase, and if they do it from the mitochondria, caspase 9 is the required initiator caspase. So that's the model. So how do you prove that beyond the shadow of a doubt? So we collaborated, well, first we made a mouse that lacked Back. That's Tulia Linston, who's sitting over there in the lab, and it had absolutely zero phenotype. And Stan Korsmeyer's laboratory made a mouse that lacked Bax, and it had essentially no phenotype. As I remember, they cleaned something out of it, but there wasn't much impressively done. Apoptosis was pretty normal in every paradigm you could do. So out of desperation, with Grant McGregor, we bred the two together and got animals that lacked Bax and Back and solved the chicken problem. Because mammals make a pad for their hands and their feet exactly the way chickens and birds and ducks do. But when they don't have Bax and Back, they keep all their interdigital webs, and they're really good swimmers. Not really good hunters for food. They didn't do well in the cages. In fact, this is a 7-month-old animal, and to get the food pellets in the cage, actually has to break down the membranes so that they can get their fingers on each of the food pellets. And that's actually why it isn't as impressive as they are when they're born, but it's hard to get a picture when they're newborns. And they have profound abnormalities in cell number homeostasis. So in the hematopoietic lineage, which we turn over all the time, and this is just a demonstration, you're not able to turn it. You have a viral infection, you accumulate a huge number of lymphocytes, and you can't get rid of the excess because there is no Bax and Back to affect apoptosis. So the spleen becomes tenfold enlarged, and just a single copy of either Back or Bax restores them to almost completely normal. So these are two redundant genes in the vast majority of tissues, one copy of which is almost sufficient to restore homeostasis. And so this is one of those true examples of redundancy in evolution, because this decision of cell death is so important to the successful development and hooking up of different peripheral tissues as we've interpreted. These Bax and Back deficient animals, when they were born though, gave us a profound ability now to start to understand various questions about what the intermediates in the biochemistry of cell death is, what proved that BH3 proteins that many people had proposed what their function was, figured out what the other BCL2 families were, and I'm just going to show you a couple of slides about that. So first of all, because this has actually become really important again in the days of CRISPR. You take something away, you got to be able to add it back and show that that's all that's wrong with things. So, and these are just actually preparations from those spleens I just showed you. So these are just B and T cells. I don't know whether this was all of them or B or T cells. We've done it in all varieties. Placing the cells just into culture. And what you see is normal B and T cells. This is why apoptosis was a big favorite of people that worked on thymic development and B cell development. Every cell dies in 96 hours, and then, even though they're in complete media. Every cell dies. But the animals that lack backs and back, not a single cell dies. So those knockouts really recapitulated the cell death phenotype that I've talked about. More importantly, if by introduction, by just transfecting in a gene, a single copy of BAC, or the equivalent of a single copy that would be in wild-type IL-3 dependent cells of either BAC or BACs restored, completely restored cell death in that paradigm. So all they lacked was a copy of BACs and BAC to which to execute themselves. So you could fully restore that. So then you could ask this question of what do BH3 proteins really contribute to? Are they death effectors by themselves, which some people proposed, or are they actually just mediators of this transition of BACs and BAC? Does their inhibition of BCL2 make a difference? Right? Because they bind to BCL2 and seem to inhibit its function, or BCL2 sequesters it away from BACs. And the result was very clear cut. If you take BACs BAC deficient cells, which are the ones in black, you add a molecule that Andreas made famous, one of the BH3s from BIM, the smallest form, and you introduce that into cells, it activated BACs and BAC, everybody died. But without BACs and BAC to activate it, nobody cared how much BIM you induced by transfection or by cell injection. So the BH3s are activators of the death process of BACs and BAC. So now we've got an ordered thing. BCL2s are inhibitors, and in the simplest Occam's razor, they sequester BH3 proteins as they're accumulated from stress pathways by either new transcription, new translation, or post-translational modification to become activated as BH3 proteins. They sequester them and therefore change the threshold of BH3 proteins that are necessary to activate the pore-forming abilities of BACs and BAC. There were two problems with that when we first did that. The one was the dirty little process. These animals are normal at birth. I mean, you can't tell that's not a normal mouse. That mouse is actually 7 months old in that picture. So the first paradigm that got the field shocked is you don't need apoptosis to live to be an adult. Now, you have different cell numbers in every lineage. So you have half the number of neurons, twice the number of neurons, but they're half the size on average for some of the talks we're going to hear in the neuroscience talk. There are many more lymphocytes, but they all turn over in normal ways, just at a tenfold higher excess. But the animal's actually an animal. Hunts food, flees danger, does everything. And the other thing, and we've had these mice now since 2001. I don't think we're actually breeding them anymore. We've finally sort of stopped. But in the 11 years we were actively breeding them, we never saw one of those animals ever get a tumor. It's not a meaningful step to tumorigenesis. Losing apoptosis doesn't make you prone to tumors. And that we still don't understand the explanation. So that, first of all, said, well, if you could inhibit BACs and BAC, it wouldn't be organismal lethal. There's still this idea that tumors select, particularly the B cell lineage, for high levels of BCL2, so there's still a chance there might be a tumor. But it also said there must be other forms of cell death. So this was a turning point in 2001. Everybody until 2001 was focused purely on apoptosis. And now you could start to make sense of other forms of cell death because we could completely eliminate what everybody agreed was genetic apoptosis or by the absence of BACs and BAC. So just going to give you a couple of teasers. Chemotherapeutic agents were supposed to induce P53 to induce these BH3 proteins that Karen and her colleagues and others identified, Noxa and Puma. They were activators of BACs and BAC. That initiated caspases, BCL2 opposed it. That's how chemotherapy works. We can get safer and more effective chemotherapy. That was the idea. And if you add a drug like staurosporin, the back deficients really helped you, right? So staurosporin inhibits growth factor signal transduction as a negative regulator, as a drug, and you can see when you add staurosporin, every cell dies, even if you lack p53 or you're wild type, but when you don't have backs and back, no one cares. Just like a growth factor withdrawal, you can do this with a chemical inhibitor and it goes away. But if you use the most common chemotherapeutic agent, an alkylating agent, nitrogen mustard, that's been activated, and now it makes no difference whether you have apoptosis or not. Absolutely zero. Cells died just as quickly, so people didn't really offended by that back in the day. So you can do this actually in whole animals. You can go back and use the Bob Weinberg first carcinogenesis cooperativity experiment where you introduce into fibroblasts Myc and RAS, fully activated retroviral forms. You get very potent sarcomas on the wild type animals that you see there as these masses. You get very potent tumors in the backs and back, which you think, if anything, they grow bigger and larger. And the p53s are somewhere in between. So very nice, and then you have control animals that are injected just for the treated, and then you treat them, as Bob did in those original experiments with a chemotherapeutic agent, cyclophosphamide, and most of the normal animals, this is his original experiment, die, except for this one whose tumor starts to grow again. But when we do it, and every time we've ever done it with back deficient cells, fibroblasts are back deficient, not the animals, these are just xenografts into nude mice, every tumor is cured. Every single one. We've never seen a tumor grow out. p53 somewhere in the middle. Most of them get cured. That's what chemotherapy does. Most tumors are p53 null. Most tumor cells are actually resistant to apoptosis by some mechanism, the best being B cells, as Tony and others have shown us. And so that said, there are other forms of cell death. This is one we said was programmed necrosis. We demonstrated that, in fact, the importance of why everyone dies is because when you have necrotic cell death, you inactivate the innate immune system. That gives the attention to the specific immune system if it's there. And that is one of the bases for why immuno-oncology works. Necrotic cell death wakes up the immune system. And this form of alkylating chemotherapy doesn't depend on apoptosis. Apoptosis is used, but there's enough inflammatory disordered death that it makes up this pro-inflammatory part of the innate immune system. So this was the first form of programmed necrosis that people proposed. We now have ferroptosis, we have necroptosis, we have many forms of induced necrotic death which act as a signal to the host that something has gone profoundly wrong and the host has to pay attention, as opposed to apoptosis where the host can effectively ignore that. And so that's one reason why we have a whole day on aspects of this form of death here at this meeting now and why it's cell death, not just apoptosis. At the same time, people got excited from developmental models about autophagy, which could clear out cells because cells would basically eat themselves to death before they induced apoptosis. And one thing that happened in this growth factor paradigm when we had cells that lacked backs and back is we could ask what happens to those cells when we withdraw the growth factor. They haven't died because they don't have backs and back to commit apoptosis. They live for another 2 weeks. What do they look like? And so when we looked at this, not having any idea what they'd be, here's what the cells look like. In the presence of IL-3, they look like this, but a week without IL-3, they are just all phagolysosomes. These cells use autophagy as a survival mechanism. They don't know how to execute themselves, so they turn to the most ancient, mostly survival pathway, which was macroautophagy, and eat themselves. The problem with that, to get energetics going, the problem with that is there's only so much you can eat before you run out of things. And so ultimately, this is another form of necrotic cell death, and it's this form of cell death that allows the back animals to maintain homeostasis of their lineages as they go through life. And so a great deal of work has been done. Nobel Prize, for justified reasons, had nothing to do with this experiment, was one for the identification of autophagy, but our cells have it too, and they use this in various paradigms. In fact, cancer cells use it in response to chemotherapy. So here's a human B-cell lymphoma line treated with cyclophosphamide. These animals had a xenograft. They all go away, but then they all come back in 2 weeks. And if you inhibit the ability of autophagy to work as a survival pathway by adding chloroquine, there's a great delay in their recovery, if not completely. Some of the animals actually are eliminated. Nowadays, about 20%, and there's a variety of clinical trials around that. So there's the big debate. The developmental models say autophagy can be a cell death mechanism. The mammalian chemotherapy experiments say it's a survival mechanism, and the truth is somewhere else, and so we'll have sessions about autophagy at this meeting. So I would really argue this is our current frontier of sorting out when apoptosis, when various forms of necrotic, and various forms of autophagy, both specialized and extra, actually contribute to death versus cell life, and that's why this meeting continues to go on. But I wasn't charged with that, so I've got about 10 minutes left before I have to turn this over to question and answers. I've got to be the setup guy for the next talk. So, we really liked the idea that cells, cancer cells, did have a crutch. There were at least some that we could identify, because what Carlo Croce had identified in follicular lymphomas in the BCL2 gene. So that meant that if we could sweep that crutch away, we might have an effective therapeutic using apoptosis to treat cancer. And so at the time we got these structures, the very first time of the BCL2 family, and started to understand how BH3 proteins interacted. We studied the structure of BCL-XL with a BH3 protein in its domain. Steve Fesick had invented a new technique for drug discovery that could potentially interact between proteins whose only real function was to bind to each other as protein interfaces. And that led to a drug discovery program that lasted almost 10 years, that led ultimately to the report of the first preclinical candidate in this field, which was Abbott 737. And I'm just going to take you through very quickly. The idea was to interfere with the holding function of BCL2 and BCL-XL for BH3 proteins. So even if they were in high concentration, and some cancer cell lines at that point were shown to have high levels of BCL-XL, others, because of the follicular lymphomas, to have high levels of BCL2, that holding function was what prevented normal apoptosis from going, because every cell line had some copies of Bax and back, and we just had to interfere with that holding function by blocking the BH3 binding domain. And so that was the idea. It was something that Tony has been the pioneer of for the last 10 years. We came to appreciate this idea that most cancer cell lines, because they were being driven by oncogenes and tumor suppressors, were under a form of what everybody came known as oncogene-induced stress. So they were more likely, the reasoning went, to depend on BCL2 family function than other cells, and that would be the therapeutic window of opportunity. And Tony has spent a lot of time showing that that could be a valid way to identify potential targets over the last 10 years. We liked the idea. We played no role in contributing to it, I should say. So here's the structure that we published in '97 of BCL-XL, which had its two central helices disappear into the molecule right here, down into the center. But there was this big hydrophobic cleft across the front of it, which had an amphipathic helix top, which is these basic and acidic domains along either ridge, and these deep hydrophobic clefts into part of the molecule, which is where we thought there might be anchoring, and that's exactly where the BH3 domains anchor to the protein as a helix. So that led to the stapled helix approach that you're going to hear from Lauren and from others, that you could make a helix that would swamp a system and initiate apoptosis. But it also led to the idea of Steve's that maybe we could interfere with this binding, but this is a protein-protein interface. And to bring this idea, because Steve and his colleagues at Abbott had just come up with a theoretical idea, if you had two hydrophobic pockets, about how you might find a drug that bound specifically and selectively to an interface. And the idea was you could use NMR and the known structure of something like BCL-XL to screen small pharmacophores for their ability to affect the local NOEs that are present in the structure and therefore perturb the pocket. And so doing that, you'd screen first for something that bound to pocket 1, a small pharmacophore, then you'd screen a second time for something that bound in the second pocket, and then you would get a chemist to link them up so they were structurally constrained, and therefore the combinatorial specificity would allow this to work. So it was the combinatorial specificity, just like VDJ recombination gives you a more complex antibody than the single number of units. You'd get that kind of specificity. It was a theoretical idea, and so Steve and his team at Abbott decided to take that on for real. They found, I'm only telling you the story because it really happened, here's the lead pharmacophore for one pocket, here's the lead pharmacophore for another pocket. Now this sounds simple. That actually only took about 6 months to find those two. But then you actually have to, and so again, they blocked into the deep binding pockets of a BH3 helix, so they would displace it theoretically, but these have very low binding affinities. And so you end up having to get a series of 5 chemists who work for 5 years to find a way to sterically constrain these two pharmacophores so that they would fit along the helix and bind into each of the 2 pockets and be sterically restricted for each other. And that drug became Abbott 737, a proof of principle in animals that you could interfere with BCL2 protein function. And then people simply, well, this is just some from pharmacology. Our original concept was to get a broad-acting one that could inhibit the 3 big family members that were thought to be active as an apoptosis time, XL2 and BCLW. Nobody ever found one, at least in our series, that had MCL1 activity. So that was always a separate domain, and many people here have worked on what's different about MCL1 as an anti-apoptotic protein. 737 bound really tightly to XL and to 2, so it was a broad-acting agent, and allowed us to go into cell lines in the NIH-60 panel, and that's where you get your first hit in xenografts. So this is a small cell lung cancer xenograft that was widely used. If it vehicles in a xenograft, it grows very nicely over this period of time, over 2 months, when placed as a xenograft. And then in a dose-dependent fashion, as you added 737, you got slower and slower tumor growth until the point where we gave 100 milligrams per kilogram to these animals, and almost 80% of the animals were cured of their xenograft once established. And this wasn't just a one-off. If you took all the NIH panel small cell lung carcinomas, you got largely the same result. 9 out of 10 CRs, 7 out of 10 CRs, 6 out of 10 CRs, and now 9 out of 9 CRs. And we had a proof of principle that interfering with BCL2 family, because there wasn't significant toxicity of this compound in mice, although you'll hear why that might have been an overstatement in the next talk as we go forward. And everybody was off to the races. So we ended this with the take-home message that BCL2 proteins might be effective therapeutics of tumors experiencing what Tony produced is oncogene-induced stress. The drug community, the pharmaceutical community really didn't like that idea. At that point, we were into precision medicine, and they said, we're not going to develop this any further until we actually find a tumor that has a specific mutation in either BCL2 or BCLXL. And that's when it takes the next 10 years and Andrew's story to get these things to realization, and I'll turn it over to that. I'm going to take 1 minute I don't earn, although I think I have 1 minute left to get 5 minutes of question and answers given when I started. I like to give an explanation of why we love coming as a lab to this meeting, but we actually don't play in cell death anymore. We realized that there's a growing community here and elsewhere in the world that were really interested in cell survival based on gains of function of BCL2 and BCLXL. Lots of people here have contributed really fundamental work on the loss of function of backs and back, well beyond what we anything did. What we became excited as is, what does the growth factor provide as a signaling paradigm that regulates survival independently of backs and back? And what does it do so that it renders BCL2s unnecessary under most physiologic conditions when you knock them out? And that turned out to be signaling through the PI3 kinase and AKT pathway. And for us, that led to metabolism and the role of metabolism in keeping mitochondria healthy, and from there, those struggles. So we really like that some of you have bumped into metabolism as part of your cell death community. We do think it's the underlying paradigm that led to both survival signal transduction through nutrient transporters and other things, and the importance of BCL2 to be modulators of how quickly you make decisions of life and death based on nutrient availability. And so that's where we are. But the real future of this field is to deconvolute all of this. But it's been a real privilege to at least give you a little history of how this meeting came about. It's thrilling for those of us that were here back in the days when it started to still see people coming, exciting, and see how much excitement there is on these other forms that are now being recognized of necrotic cell death, autophagic cell death. So with that, I'll stop and take any questions, and really appreciate your time and the invitation to be here today. And this is the time for all the oldsters like me to correct the record for everything I misspoke, which I'm sure I made it simplistic to get it done in 45, 50 minutes. Somebody's got to be brave. Andreas is always brave.
So I just want to correct you that when you said that blocking apoptosis doesn't cause cancer. So we've had back double knockout mice around too, especially mice where we just gave wild-type mice a back double knockout lymphoid system, and a given percentage of these animals gets plasmacytoma. So
Right.
certainly blocking cell death can lead to cancer, admittedly only after other oncogenic mutations come in.
Right.
BCL2 transgenic mice do get a low incidence of lymphoma that is
Right.
highly significant over the control that are made. So I wouldn't go as far as saying blocking apoptosis
Okay.
might cause cancer.
So I don't think, which is why Andreas and I always have these fun arguments, I don't think I said that blocking apoptosis doesn't cause cancer. I said our mice never get cancer. That's not the same signal, okay? Now, it's absolutely true that Stan Korsmeyer's lab showed introducing a BCL2 oncogene could, over a long latency, give a higher incidence of cancers, mostly of the hematopoietic system. Andreas has published a number of examples when a driver oncogene is present directly or indirectly because there are a large number of endogenous retroviruses in certain strains of mice. There's no question that anti-apoptosis contributes to the higher incidence and the better take of initialized tumors. There's absolutely zero question. Hundreds of experiments to say that. The striking thing to us is, when we did the experiment, we were sure we were going to see hundreds of really cool tumors we'd be able to characterize, and we're still waiting for the first one out of those mice, and we gave up after 11 years. That does not mean that others breeding different pairs and different combinations. This is an outbred mutt in these animals. Most of the time you try an inbred back doubles, it's embryonically lethal.
Why, can you conjecture why it's relatively common to find cancers expressing anti-apoptotic proteins at much greater levels than normal cells? I don't want to say overexpressed, I don't know how to, but anyway, they got a lot more of that. It's really rare to see a cancer cell that has lost backs and back. You can find the rare cell line, but it's really rare.
So this is really important. So there's a famous story about Harold Varmus when he and Mike Bishop won the Nobel Prize for the discovery of SARC as a retrovirally captured oncogene that has just been mutated enough to be constitutively active and to create a signaling paradigm. And the way Harold was going to prove that this was such an essential pathway was to knock out SARC. And some of the older people probably know the answer. You've never read a paper about a SARC knockout mouse, right? Nobody here has ever read one, because it has an absolutely normal phenotype. And so Harold's only explanation was, well, that's because SARC's so important, it's multiply redundant, so there's other intracellular tyrosine kinases. That actually turns out not to be true for SARC. They do get osteopetrosis, it's just nobody was smart enough to look at all the bones. They had much heavier nose, and if you do a test of the mice, they actually sink because their bones are so dense, they sink, and so you can tell a SARC knockout instantaneously by just putting them in a swim tank. So it's not redundant. We just couldn't figure it out. I think backs and back really are fully redundant. And I think it's so important to regulate rapidly, since we're a modal animal that has to hunt for food and flee. You've got to have the ability to eliminate cells that aren't going to keep you alive as quickly as possible. You need it because we're placental, and so mammals in particular have to have it because you got to get through the birth canal, so you can't go through with 50% more neurons or you're screwed, because your brain will get squished, which was some of the phenotypes of the first knockouts that we know. But it really says, I think, that apoptosis is a centrally important development and homeostatic mechanism. The interesting thing about it is it's only a secondary, and so Andreas and I do not disagree at all about what he just criticized me about. As a secondary pathway to infer better survival of what has been called, in the broadest sense, so we could have a long discussion about oncogene-induced stress, it is the most potent way to synergize with an activating oncogene we've ever seen. Is that fair, Andreas? Okay. We agree. We come at it from a different point of view, but we agree. So I really think that something you've been an important purveyor of is that oncogenic stress. So that can initiate from the ER. And there are transcription factors that activate the expression of BH3s for death, but they're also, which people don't think, as you survive ER stress, you upregulate BCL-XL, its expression a great deal. So that's on a transcriptional basis. So I think there's adaptation to the stress that's based on the oncogenic transformation, the specific oncogenes transferring a tumor that activate adaptive pathways that turn up the expression of BCL-X2 because they've made the critical decision they're going to live and not die. And so just as we get excited that there's pathways to activate BH3 proteins to execute you, cells are exploring survival pathways that allow you to get at that. And I think we're going to see that. Judy's not here that I've seen, but I think the senescence stuff is really getting at that we're going to see tomorrow morning. I think there really is this a real redundancy. Some cells should just never divide again, but they need to cover a barrier epithelium, and that's a senescent cell. Senescent fibroblasts, senescent other things. And I think they've upregulated a lot of survival pathway, overexpressed them. Cancer cells have figured out how to do that through transcriptional mechanisms, while at the same time retaining the ability to have driver oncogenes that drive the cell cycle progression. Yeah?
I mean, you've nicely shown these experiments of BAPs being able to act as a calcium channel, basically. So what's your take on BCL2 family proteins and calcium flux, for example?
Yeah, so I took out all the slides. We published a number of papers about calcium flux.
I mean, there are tons of calcium channels out there. So
Yeah.
why would you...
But I think the one thing people don't think about is that calcium creates an electrogenic problem that has to be solved by somebody. And so I think these are compensatory channels. These basically solve the voltage problem. So calcium are all uniporters. You can't get very far with a uniporter because you create too big an electrochemical potential. You've got to have something to dissipate the charge that's created. So I see them as safety valves, pressure outlets. And I think BCL2 and BCL-XL function particularly well as safety valves. So they're there to keep voltage-dependent anion channel, as we've published some studies, open and in the open configuration, because it's a voltage-dependent anion channel. The minute voltage appears on the outer mitochondrial membrane, it closes. And then you can't get anything in and out of a mitochondria. You put BCL2 or BCL-XL next to it, and under all conditions, voltage-dependent anion channels continue to exchange ATP and other things that are critical for survival. So I think most of the time we think about them as compensatory regulated channels, just the way the pore-forming proteins, but now in a positive adaptation to dissipate potential. And that's where you do need to dissipate potential, where you release calcium at the ER, where you actually have to exchange charged ions to be able to maintain APP production at the outer mitochondrial membrane. This is not the integrity of the inner mitochondrial membrane. This is the outer mitochondrial membrane that's in exchange. And so we think, and I've been saying this for now 15 years, all of our data fits a model that BCL2 protein is an umbrella. It's opening and inserting into a membrane and creating a function. Okay? We entertain two ideas. One is that it's a charge dissipator, like I just said. The second is that it's a regulator of cholesterol metabolism by regulating the distribution or insertion of cholesterol into the membrane. Because as an arm, it just does this. And in our hands, because the soluble structure of BCL-XL, the one thing that crystallizes with it is cholesterol in the cleft, is that that's what it does. It's an insertion domain. It takes something from outside, grabs it, throws it in. This is the alternative from the thing. Never done anything to disprove it or prove it. We've tried a bunch of things. And BCL-XL is always doing this and releasing here. And backs and back are doing exactly the opposite. They're grabbing here, releasing here. Same function, different sign. Add a BH3 protein that forces you here, and you activate the bad function by dispersing cholesterol locally for backs and back. Keep you stuck here as BCL2 binding a BH3 protein, and you can't reinsert any more cholesterol for stabilization. So we think it either perturbs local lipid concentrations or provides a safety valve to voltage. Both of them work in the model I just said. We tried very hard for about 5 or 6 years to try and prove either one right or wrong to our satisfaction, and we've not been able to do either. So it remains, to me, a theoretical idea, but it's going to take people better at biophysics than we were. And we've moved on to the metabolism issue. But it's a great question, and I'll stick there until somebody proves us wrong or right. Thanks. Thank you very much for having me.